Don't just give me my own notes back. Okay. Then let's see. So this is 7A. Cool. So it says in a series of diagrams, I need my highlighter. In a series of diagrams, give us the process of transcription. Okay. Now, be sure that you read these next couple of exams, both in 7A, 7B, and then when we get into Core 8, 8A, and 8B. It's really important that you read the question, okay? And I know that sounds kind of silly, yet it's important in that when you're taking any standardized test, whether you take the IB exams, the AP exams, when you get into a standardized test environment and that adrenaline starts to flow, sometimes we get sloppy and we don't read the questions, okay? So IB will not always ask you transcription and translation. They may just ask you one. And so if you glance at it and it's really asking you transcription, and you provide them with this beautiful explanation of translation because you didn't read the question, okay, they're going to award you a 0 because you didn't answer the question, all right? So please, please, please be sure when they ask you, and they will, prokaryote, eukaryote, plant, animal, meiosis, mitosis, DNA, RNA, photosynthesis, cell rest, transcription, I mean, just classic questions, but they will only ask you one of the pair, all right? Pay attention to which one that they're asking. So we're having a series of diagrams. Now, how you decide to break down transcription is up to you, okay? Just because the diagrams that I drew, I might have taken 3, 4, 5 steps. The textbook might take 5, 6, or 7. It's whatever you think is necessary to show initiation, elongation, termination. Okay, but to show those 3, that means you need a minimum of 3 diagrams. Okay, so 3 or more. If it takes you 4, if it takes you 5, you see somebody else's exam and they took 6, fine. Okay, I'm looking for you to diagram initiation, elongation, termination. Diagrams, just a series of them. Then, since I'm grading these electronically, usually, as you know, you're junior, I've got, a good old green pen, and I track where the terms are. That helps me track in my head the vocabulary that you use. For me to highlight them electronically and put a check mark on them will triple my grading time. Okay, so you can really, really, really help me out. Underline the terms, please. Please, you can help me out with time. Okay, and then these terms, for the most part, these are a little mixed. I used to do them alphabetical, and it would really turn the students inside out. And so I think this is the best order. I'm trying to do it in the order that they will occur, kind of. And if there's any terms in there that don't make sense, just ask. Okay. Then question 2, again, a series. And here we're also talking about translation, which involves, ready, initiation, elongation, termination. So you need at least 3, right? So then it says, including the A, P, and E sites, don't forget that. You are not drawing me the freshman and junior ribosome. That's only 9, excuse me, it's only 3 bases wide. I was trying to get you comfortable really with just the P site, right? And you guys are freshmen, and even when you're sophomores, all I'm teaching you is the P site. Because if I can get you solid and comfortable with the P site, then it's a charged tRNA in the A site and an uncharged in the E site. But conceptually, you understand the heart and soul of that P site with translation. Okay, so you're drawing me the real size ribosome, 9 bases wide. Okay, let's see. As you explain this process course, help me out. Underline the terms. Okay. And then there are a number of terms here. So I strongly recommend that as you go through these, right, check them off, circle them, scratch them out, box, I don't know what you want to do, but be sure to check these off, okay? Usually the only place that students miss points here is because they don't show me the terms, okay? And I'm like, where's the release factor protein? Or did you actually label the stop codon, right? So be sure that you go through this and label, especially there's 3 of them 4 of them, let me think here. So there's initiation, elongation, termination, translocation, and the polysome. Okay. Now, if I were you, I would do the polysome as a separate diagram. Okay. If you want to give me a highly detailed messenger RNA with multiple ribosomes and really get into this, if you want to do that, go for it. Okay. I were you, make your life easy. And after you do the whole initiation, elongation, termination, then go underneath of that and keep this diagram simple. Right? Give me 3, 4 ribosomes, or they're getting skinnier the longer that I draw them. That's interesting. Show me a small protein, medium protein, longer protein, and almost done protein. Okay? And then when you show me the polysome, be sure that you bracket or circle the whole thing. Students will then, they'll miss points because they will come in here and they'll go like this and they'll go polysome. I have to mark that wrong. That's not a polysome, that's a ribosome. Okay, so that is incorrect. When you do the polysome, it's the whole complex. Okay, it's multiple ribosomes all reading one messenger RNA all at the same time. Okay, your diagram for the polysome can be this simple because you've already provided me all of the details on these diagrams up here. I'm not going to have you give me this really detailed diagram down here because then I'm asking you to draw it twice. And I think that's mean-spirited. Okay. The only other thing that I consistently see as a mistake, where's the release factor protein here? There's no tRNA in the stop codon. Okay, I will students bring in a tRNA on the stop codon, and then they will attach an release factor protein to the tRNA, and I'm like, that doesn't exist. Okay, so when you show me that stop codon, and you have the stop codon like such, then speaking of green pen, the release factor protein comes into release factor protein. It comes into that like such. Here's your ribosome, like such. The release factor protein actually comes into the A site, like such. Okay, so there's no tRNA in the stop codon at all. The release factor protein comes into that A site and causes that whole complex to dissociate, to separate. Like teens, when you walk up and say hi in the hallway, and they separate. So 7A comes down to transcription and translation. Any questions on that?
So far,
Pardon?
no.
Good? Okay. All right. 7B. 7B, same thing, is step by step. Okay. Now for the replication fork on 7B, put to me where show me the same length of it. Okay, and then open it a little bit, and then more, and then almost done, and then complete. Okay, so show me a minimum of 4 diagrams here. Okay, but it is the same length of DNA. So open it up just a little bit, and then open it up some more, and then almost completely opened up, and then oh boy, I'm going to have to really squeeze this in, and then complete. So here's a little bit, fragments, total. And then maybe you join those two together for your DNA ligase, and then to complete like that. You see how these 4 sections of DNA are all the same length? Okay, show me the same thing and open it just a little, and then more. Okay, so here is your initiation, right? Then here you're able to show me the Okazaki fragments. Then here, able to show me the work of DNA polymerase 1 and DNA ligase joining the Okazaki fragment and your completed fragments. It's a concert, Okay, so again, it's a minimum of 4. Some of you will draw me 5. Some of you will draw me 6. Fine. Okay, I would do a minimum of 4. Okay, and then always, 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 what goes here in this top right corner? Always. Which end is that? 3. 3, you bet. Okay, don't waste your time thinking 3, 5, 5. It's anchor yourself in that 1, 3, and then from that 3 you can figure everything else out. Okay, but that top right is always anchored in the 3. Otherwise than that, I mean, these questions are kind of straightforward, I hope, right? Show me step by step, help me out by underline. Replication fork, a series of diagrams. I would do a minimum of 4, right? Discuss the process, underline the terms. Now, for the color coordination, since you are creating your own diagrams, if we were in class, I would hand you a box of crayons, right? Some of you have crayons, some of you have highlighters, some of you don't. That's totally cool, okay? Even if you have a pencil, then take your enzymes and put different patterns in them, right? Make one of them empty, make one of them diagonal, make one of them horizontal, color one of them in, right? Make another one, and I don't know, make it polka dots, right? Like such. So don't get too hung up if you're doing these by hand and you don't have colors. Even if you have a pencil, it's totally cool, or pen, right? Use patterns on the enzymes to show me DNA, or let's see, RNA primase, DNA polymerase 3, DNA polymerase 1, DNA ligase, helicase, right? But give me some kind of difference, whether it's color or pattern. Totally cool, because obviously I can't hand you crayons and different colors. Cool?
Yeah.
All right. Then for number 2, we're going to talk about good old AATS, right? I need one diagram that shows the general role of the enzyme, and then a second diagram that shows me the specific mechanism. I want you to step back and give me this diagram is the forest, right? This is the big idea. I want you to be comfortable with how this enzyme functions on the grand scale. Okay, do you understand that we eat food, and that food then is digested or hydrolyzed in your stomach and your intestines? Those amino acids then move into your blood, your heart beats, and then pushes that blood with the amino acids through your circulatory system. The cells absorb the individual amino acids, and this is the enzyme that couples a specific amino acid with a tRNA. We call that then a charged tRNA. Okay, I want you to give me the big idea. Then here, I want the trees. Here, I want the details. This is the nitty-gritty, where you have the enzyme with 2 areas to its active site, right? The area on top gets the ATP and the amino acid. The area on the bottom bonds to a very, very specific tRNA, okay? And this enzyme, of course, has that kidney bean shape to it. That's why I'm doing this, okay? Same thing, please create these yourselves. Have this go through your brain, whether it's with pen and pencil or whether you do it on whatever graphics program, drawing, whatever you decide electronically is totally up to you. But please don't give me my own notes back, and please don't give me the book notes. Copy paste does you no good because it doesn't go through your brain. Okay, and you've seen my hand drawings, so these don't, it doesn't have to be super high tech for you to understand the concept. Okay, all right, then number 3, synthesize a protein with 3 specific and different amino acids. Okay, when you were juniors and you were going through core 2, and we synthesized a fat and we synthesized a protein, when you brought those amino acids together, and you did, of course, the central carbon like such, and then the carboxyl group on one side, and then the amino group on the other. As juniors, we said, hey, just put R1, R2, R3 here, and we'll get to the specifics later. Hey, guess what today is? Today is the specifics. Okay, so please go to the image. Have I posted that? You can Google that. Look up the structure of the 20 amino acids and pick 3. Okay, I want 3 different amino acids. Okay, I have to add that different, otherwise people pick the first amino acid where the R group is just hydrogen, and I'm so sick of that amino acid. Okay, so please give me 3 different, and then bring 1 amino acid in, bring 1 amino acid in. The hydroxyl group, or the OH off of one group, the H off the other, leaves as water, and then the carbon and nitrogen bond. Okay, so the list here should be pretty straightforward, because we're returning back to core 2, because I want you to understand that on the ribosome, where we show 2 spheres, 2 circles just bonding together, I want you to make the correlation that each sphere is one of those, with that central carbon, the amino group, and the amino group and the carboxyl group. Okay. You should have your core, let me think, this is either 2A or 2B exam. I think it's 2B. It's one of the two. Okay. Then also going back to core 2, tertiary and quaternary protein structure. Right. So this is where in the tertiary protein, it folds back on itself, makes a globular form. Right. Then all a quaternary structure is multiple tertiary structures bonded together. Maybe get a little of this over here, then maybe a little of this over. And sometimes there's as many as 17, 19 tertiary proteins all bonded together to make this gigantic quaternary protein. Okay, again, because I want you thinking ribosome, primary protein. That primary protein then leaves, starts to fold and twist, alpha helix or the beta pleated sheet. Right, and then that starts to twist and fold on itself even more to make the globular protein. My email needs to go away. Like that and like that. Okay, now why am I taking you back to these proteins? Because there's a question that IB loves to ask. They love to ask you about sickle cell anemia. All right, and there's the reason why. It ties together genetics. It ties together protein synthesis. It ties together human anatomy and physiology. It ties together the folding of proteins and how they're important. So IB has this much content to test you, right? But they can only ask this many questions. Okay, so they really, really like to ask this kind of question because they hit multiple topics all at the same time, all right? So there's a really, really cool image in your book. It's image 522. And we hit this when you were juniors, when life was simple. Right? Remember this image a long time ago when you were young? Maybe? Maybe. Okay. This is a classic question on the IB exam. They really like to ask this question. Okay. So let's be sure that we know what we're talking about here. We're talking about sickle cell anemia. So let's hit the genetics on this first, and then we'll go through the biochemistry of it. So let's see. So that is sickle, KLE, right? Yep. Sickle cell anemia. This is an autosomal, which means it's carried on one of the chromosomes 1 through 22. It's an autosomal recessive genetic disorder. Okay, you don't walk down the street and go, "Oh, I caught sickle cell." Okay, not the way it works, right? You carry these alleles in your DNA. Okay, so since it's recessive, big A is for typical hemoglobin, and we'll talk about hemoglobin in just one second. and little a is a misformed or misshapen hemoglobin. In fact, what the hemoglobin forms is it is linear. Okay? Hemoglobin, who cares? What does it do? Who cares about hemoglobin? What's the big deal if my hemoglobin doesn't work properly? What's the job of hemoglobin?
Transfer oxygen.
Yep. You have your lungs. The lungs to the tissue back to the lungs. Okay? The lungs deliver oxygen to the tissue. The tissue uses the oxygen for cellular respiration. The tissue then releases the waste gas of cellular respiration, which is CO2, and that returns back to the lungs. Okay? You then have the protein, hemoglobin, which actually bonds to these individual gas molecules. But the hemoglobin is then carried inside of a cell. And it's a blood cell. And when you put this blood cell underneath the microscope, it's red. So what do you think we call this blood cell that's under the microscope that's red?
A red blood cell.
A red blood cell. That's why we call them red blood cells. So what a red blood cell is it's just a bag of hemoglobin. That's all it is. There's not even a nucleus inside of the red blood cell. Okay? It's just a bag of hemoglobin. And so the red blood cells show up to the lungs and drop off carbon dioxide, pick up oxygen. Heart beats, right? Blood leaves. Blood leaves to the tissue, drops off the oxygen, picks up the carbon dioxide, heart beats, and this cycles back and forth. Okay? So if someone's hemoglobin is misshapen, they have a difficult time transporting oxygen. So individuals with sickle cell anemia have difficulty transporting oxygen to the somatic cells. All right, who cares? Why is oxygen important?
ATP.
Right. Oxygen is a reactant for cellular respiration. So they have reduced reaction rate for cell resp. Well, if they have a reduced reaction rate for cellular respiration, they have reduced amounts of what?
ATP.
ATP. And they suffer from low energy levels, literally. It's called malaise. And they're tired all the time, okay? Also, the cells become a sickled shape like this. And when the when the blood vessels around corners in the joints, these sickled cells get stuck. And so they get blood clots in their joints, all right? So they have very low energy. Anytime they exert themselves, they have almost no stamina. And the sickled cells can get stuck in their joints and cause a lot of joint pain, okay? All because one little part of the protein doesn't fold properly. Okay, it's when the cell or the protein goes from secondary to tertiary shape. Super important, which is what they're showing you here. Okay, so this protein here, this is your hemoglobin. Okay, so the folding of this protein is super important. So let's highlight this. Let's call this typical hemoglobin. Now, on this diagram, if you want to use the diagram that I posted it blank. If you want to write on this or type on it and then use that for your answer, I'm all for it. If you would rather redraw parts of this, that's totally up to you. I think it's a little much to ask you to redraw this unless you have the artistic ability, which if you do, how cool is that? So this is typical hemoglobin. I didn't give myself enough highlighter. Okay, so the order of the amino acids here. What do we call this structure when it's just the order of the amino acids?
A protein.
Right, that's a primary protein. So this is your primary structure. Okay, now when this protein is first formed, amino acid number 6 is really important. Okay, so this amino acid right there, number 6, is usually glutamic acid. Okay, and since glutamic acid is in number 6, glutamic acid is charged. It's polar. Okay, and that's important. All right, because number 6, the amino acid, sits right here and is attracted to that area of the protein. So when the protein forms its tertiary shape, this area of the protein right there is polar. And you know that polar is attracted to polar. And so it takes the top part of that protein and it folds in. Why? Because it's polar here and it's polar here. And so the protein folds in and makes this nice globular protein. So what you're looking at here, this is the secondary and tertiary structure. So this is secondary and tertiary structure. It's a nice rounded globular protein. And technically, this is called the beta subunit. Okay, well then this tertiary protein gets bonded. So here's your beta and here's your beta, and this gets bonded to what are called alpha proteins, and it makes this beautiful round globular quaternary structure. This is your quaternary structure. And here is your globular hemoglobin. This is what you and I have. Nice globular structure, okay? When these globular proteins are then surrounded by a plasma membrane, you get a typical red blood cell. And it literally looks like a jelly donut, okay? It's a nice circular cell, and it even dips in the middle just like a jelly donut. You have these in your bloodstream by just millions and millions of these, okay? However, there's individuals who have a mutation, and we call them a sickle cell anemia. And I still didn't give myself enough highlighter. So you have a sickle cell anemia individual. So what happens? Get a load of this. 1, 2, 3, 4, 5, 6, 7. Do you see how number 6 is colored red here?
Yes.
Yes. Number 6 is colored red because the mutation is a valine. It's one amino acid. The 6th amino acid, instead of being glutamic acid, right, which is polar, it's valine. And guess what valine is?
Nonpolar.
Non-polar. And this amino acid sits right about there. Okay? So instead of that that polar amino acid being attracted, now this amino acid is non-polar. Well, if this area is polar and this is non-polar, what happens to these two areas? Are they attracted to each other? Nope. They're repelled. It makes the top part of this protein kick up because of one amino acid. It's a substitution. It's a substitution mutation. It substitutes valine for glutamic acid. It substitutes a non-polar for polar. Therefore, the protein structure doesn't form properly, and it has this arm out the top. Okay, so this is your secondary and tertiary structure. So this is your secondary and tertiary structure. I'll use a little shorthand here. This is your beta subunit. You then form alpha subunit, beta subunit, beta subunit. And do you see how this arm sticks out the top and the bottom? Okay. Now what happens is the hemoglobin molecules, instead of being these globular little blobs, they have an arm out the top and the arm out the bottom. And look at how they assemble. So the hemoglobin molecules interact. And they make a linear molecule. They make this structure here. It almost becomes a crystalline fiber. Okay. Well, why is that a problem? This protein changes the shape of the red blood cell. Okay, when you change the shape of the protein, that changes the shape of the red blood cell, and now you have a sickled cell. You have a sickled red blood cell. It's curved. Okay. Then, since these individuals can't transport oxygen very well, that causes a condition called anemia. Therefore, my anemia is caused by my sickled cells. So this condition, these sickled cells, causes sickle cell anemia because I don't transport oxygen well. It's a genetic disorder, it's recessive, and it's all because of the substitution of one amino acid, only one. Yes, ma'am. Correct. That's exactly right. Okay. And I'm making a big deal out of this question because IB loves this question, right? Because they're hitting polarity, they're hitting protein structure, they're hitting red blood cells and the transport of oxygen. They're even talking about cellular respiration and ATP. I mean, they love this question. Okay, so what I'll do is I already posted let me double check that. Let me be sure that I did that for you before I say I did that. Oh, there's the extended essay guide for the juniors. Start talking to the juniors about the extended essay on Friday. Let me make sure that I posted this up for you. Core materials, we're in core 7. Maybe I put it in with your exams? Maybe I put it with the exams. Aha, I did. So in with your core exams is the image that we just did, and it's blank. Okay. So what I will do now is what I just made for you here, I will take an image of this and I will post with the details. I will post this with this file. Okay. What I want you to do is I want you to annotate this. Okay. No fair taking my notes and giving this back to me. Okay. If you would rather use the textbook image, then annotate that and use that as the diagram for your explanation. Okay, or if you guys want to draw it on your own, totally up to you. I'm not going to get into the nitty gritty of the shape of the protein here. The most important thing is that arm that sticks out the top. Okay.
What is the purpose of that protein?
The purpose of hemoglobin is it's inside of the red blood cell, and hemoglobin bonds to oxygen and carbon dioxide, and it is actually the protein that's responsible for gas exchange. So hemoglobin grabs oxygen in the lungs, goes to the somatic cells and lets the oxygen go for cellular respiration. The hemoglobin then picks up carbon dioxide, goes back to the lungs, and releases carbon dioxide as the byproduct of cellular respiration. Oxygen to your somatic cells, carbon dioxide back to the lungs. This is the protein that's inside of red blood cells. And I'll post this also. Okay, so I don't think it's fair to start on photosynthesis today. I would like to cut you guys loose and give you 45 minutes to either, if you want to go work on your TOKSA, you want to work on your IA for any class, or you want to dig into this exam. Let's do what is best for you.